Why do we get sick? This is a good question for human health. This is a good question for evolution. And this is a better question for the evolution and human health lecture. Let's get going. Give examples of host-pathogen interactions in evolutionary time. Relate natural selection to influenza. Define H and N in terms of influenza strains. H1N1 and such like that. Relate antibiotic resistance to human history and trade-offs. Give examples of when it is adaptive for a pathogen to kill the host from a couple different perspectives. And relate evolution to cancer. So evolution matters to human health. Our pathogens evolve in our bodies, as we saw in lecture number 1 with HIV. Antibiotic resistance is a real and present danger, as we saw in a later lecture. Our pathogens are evolving for better transmission, as we see in COVID. And cancer cells are a population of cells that are evolving. If you were there for the Thursday section where Corinne presented her paper, we saw this. We know that we evolved from other primates, as we're going to see in later lectures, and we have evolved in the recent past. So due to our large population, no migration, low level of mutations, relatively random mating, and lack of natural selection, we're currently not evolving very much. But the pathogens that attack us are. So let's talk about those pathogens. First off, let's track some of these pathogens. Back in 2010, there was a large earthquake, and following the earthquake, there was an outbreak of cholera. So we found, by using a phylogeny, that the cholera outbreak could be traced. The outbreak of cholera could be traced to other types of cholera that were present in Nepalese populations of cholera. So we see all these Nepal populations of cholera, which they themselves had come from Bangladesh, and we said that the Haiti population of cholera came from Nepalese aid workers that had come over to help people in Haiti. So they came to help, but the cholera they brought came to infect. So that was a bit of a problem for them. We've also traced this with AIDS cases, with the case of Louisiana versus Schmidt back in lecture 1. All right, a good example of evolution and pathogens is influenza. Now this chump just wants to infect as many people as it can. Oh boy, here I go killing again. The pathogen has to keep evolving though, because a host that is infected with HIV, with influenza, calm it down there, a host that's infected with influenza is going to be immune to it in the future. And if you are immune, well then, it can't infect you again. So it's constantly reducing its own food source. It has to evolve in order to infect new food sources. This is why you can get infected year after year. So this is it's a protein coat with neuraminidase and hemagglutinin, and then a kind of a matrix as well in the coat, and then 8 different RNAs used to infect. So you're looking at 8 RNAs already, you're thinking that's a lot of variation. And yes, it does have to keep evolving. So it evolves about 1 million times faster than us. We're basically standing still as this thing is evolving new forms every year. And we'll hear about those new forms. We usually hear about them as like H, X, N, Y. So what does the H and the N mean? Well, H is for hemagglutinin. And hemagglutinin is this, it's a protein. There are 18 subtypes of it that have been identified thus far. The last one I think was identified in 2004, might have been 2012. And this hemagglutinin is going to bind to the sialic acid containing receptors on cells. When it binds to sialic acid containing receptors, that allows it to kind of insert its RNA into the cell. So unfortunately, it's also a target of your immune system to detect this. It's called hemagglutinin because if you just take a pure amount of this and put it in blood, it causes the red blood cells to clump together. So that's why it's called hemagglutinin, pretty simple. Then there's neuraminidase. So it acts as a sialicidase, so it breaks down sialic acid. Not quite clear about how it helps you infect, but it's there to help the hemagglutinin do its job and get inside you. And we look at the sequences of the neuraminidase and hemagglutinin genes, and that lets us know it's what the H and the N are. So you can combine these different H's and N's to give different combinations to make different flus, which is constantly evolving. So other components, of course, can evolve as well in there, and your body's defenses will acclimate to whatever's in the environment at the time. And we know that this is actually really not a neutral evolution. It's not neutral evolution. So the neutral theory, no, it's definitely under directional selection. The hemagglutinin gene specifically is under natural selection. If you look at the genetic divergence from the original sample in how many nucleotide substitutions are occurring in the hemagglutinin gene, you can see that it is more than one per year. There's more the substitutions going to fixation than would be expected through neutral evolution. And you can see this phylogeny of all the different strains of influenza that kind of come out, the Victoria strain, the Houston strain, Wyoming strain, Ecuador strain. So we see all these different possibilities coming out. And of course, there's always that movie Contagion, where you get a bat and a pig. They have there's some sort of a cross genetic, influence between the influenza gene. Basically, the influenza comes from the bat, infects the pig, and then from there it gets to a human, and that kind of destroys a lot of humans. It kills 6 million in the movie. I had to walk out. I didn't like that movie at all. It's a little, I don't like infections, and I don't like the flu. Flu vaccines are actually allowing the body to recognize the hemagglutinin before it infects. And the flu vaccine has to be specific every year to what hemagglutinins are around. So where does it come from? Well, birds, pigs, horses, bats to a much smaller extent, and other humans, of course. Birds can pass the flu on to pigs. It can recombines with other varieties, making new strains, so there you see an increase in variation. The 8 RNAs are heritable. You have directional selection. Ba-boom. There you go. Evolution. We have swine flu and bird flu, but not horse flu, in part because the number of birds and the number of swine that are interacting with humans. It would not be surprising at all if you were to tell me that back in the medieval times, there were horse flus going around because humans were much more interacting with horses back then than we are now. So as we change what animals we interact with, we shouldn't be surprised when we get new strains of flu. Case in point, as civilizations in China are growing and cities are growing, they are coming more in contact with bats. So you're going to hear more and more about bat flus and bat diseases. COVID-19 being one example. There's another one festering in Southeast Asia, which causes massive headaches and brain swelling and death at a much higher rate than COVID. These things can hop over between species if they're similar enough. In short, don't go hanging out with a whole bunch of dirty chickens. Stay away from the dirty birds, and don't bathe with pigs. But if you think about where these diseases come from, you can also see in the medieval times and before, humans lived very closely with animals. I mean, Jesus was born in a manger, So you see humans dealing with animals all the time, so it's no surprise when things kind of jumped over. A lot of our diseases can be traced to animals. So next, let's talk about antibiotic resistance. Antibiotics, we've already discussed this a bit in class, are a natural selection pressure on bacteria. And the resistance depends largely on the pressure exerted by antibiotics. So how much are antibiotics being used? How well are they being used? Are you taking the full dose? Are you completing the whole dosage? It's relatively safe to assume that resistance alleles will arise by mutation given enough time to any antibiotic. And we've seen it with most antibiotics already. We see that resistance correlates pretty well with how much people are using antibiotics. So the less you use antibiotics and the more you use other alternative remedies, the more you're going to get, the more antibiotics, the more resistance. Short story. So how did it become resistant? So mechanisms of antibiotic resistance, first off, you can wait until the antibiotic application is over. This is what's used by tuberculosis. What it does is it forms itself into cysts in cells, and they can actually just wait until the antibiotic stops being applied and then recolonize afterwards. And there are a lot of antibiotic-resistant bacteria out there just like this. You can also keep the antibiotic out of the cell. We talked about this with efflux pumps. And efflux pumps can basically pump that antibiotic right out. You can digest or destroy the antibiotic, as we saw with the beta-lactamase genes in penicillin-resistant bacteria. There's also mutations that alter the drug target. So if you remember, there's tetracycline will actually inhibit ribosomes. It'll inhibit ribosomes, and you can have mutations in the ribosomes that make them less susceptible to tetracycline. So by altering the target, you can, of course, resist the antibiotics. So this is a form of directional selection. And as such, it's only going to be adaptive when there is antibiotic in the environment. So why aren't all bacteria naturally antibiotic resistant? Because there isn't the natural selection for it. Now there is some variation within bacteria that allows them to be antibiotic resistant, but often this comes at a cost. So the beta-lactamase gene, if it's going to be digesting cephalosporin, third generation antibiotics, and it has to have a larger active site. If it has a larger active site, it's less stable. If it's less stable, there needs to be a secondary mutation to make it more stable, yada yada. There's a cost. And this cost to antibiotic resistance means it doesn't just continue to evolve in the absence of antibiotics. And this is where in Iceland and I think Denmark as well, you have antibiotic resistance is no longer increasing. There's not less antibiotic resistance, but it's not increasing as much due to more intelligent applications of antibiotics. So when they're actually going to give them, when they're not. By this kind of variance, you can reduce the selective pressure, thus reducing the drive for increased antibiotic resistance. However, it doesn't just go away, because there are other potential mutations to decrease the cost of resistance and allow the pathogen to evolve, being able to compensate for the cost of antibiotic resistance. In short, yes, we're all doomed. As long as you're using antibiotics, there will be directional selection. As long as bacteria have variation, they can make up for any costs. And one possibility is just design new antibiotics, which is what you can learn in medical botany, where we talk about how many different plants have things that are effective against bacteria. So there's, hashtag medbot kind of connection. What we see, though, is that it's not just going to go away because we stop using antibiotics, but we can stop it from becoming more of a problem. 1, 2, 3. Virulence. Again, sorry for all of the jumping around, but this Camtasia is being a little difficult during this lecture. Anyway, so you have viruses and bacteria, and they can actually span the gap from symbiosis of parasitism to a symbiosis of mutualism. Symbiosis just means living together, and when a bacteria and a human live together, or fungus and a fly, as in this case, they can go a whole bunch of different ways. This fungus in the fly, as you see, is parasitism. What you have is the fungus actually infecting the fly and then living in the fly for a while, telling the fly to go fly up somewhere high, and dies. And then the fungus basically liquefies the body and creates these spore-bearing structures to spread its own spores. And it works well for the fungus. Typhus parasitizes humans. It makes a human defecate more, and it spreads in those feces. It actually spreads by flies, too. Then there's commensalism, where it's a +0, and this is a streptomyces, I'm referring to the fungus type, that it can live in your body, and you may never know, and that's fine. Some of these do cause disease, but most of them just kind of live alongside you. Commensalistic. You don't even know they're there. And last, mutualism, which is E. coli. And, of course, Lactobacillus acidophilus. These, most of the time, these are going to live mutualistically in your body, giving you a benefit, and you give them a benefit of free room and board. So why choose pathogenicity? They said I could be anything, so I became a pathogen. Okay, no need for that. Some of them don't mean to be pathogenic, and that's what we call coincidental evolution. So this is the case where the organism in question, in which case tetanus here, it produces its own byproducts. But it just so happens that its metabolic byproducts are very harmful, even fatal, to humans if these bacteria are introduced into a human. Now, tetanus never wanted to be in a human. It just wanted to chill on some rusty nails deep in the soil all by its lonesome. But it got into a human, and it just so happens that it is capable of living in that anaerobic, iron-rich environment of a human body. It produces these byproducts, and these byproducts just happen to be lethal to humans. Many new infections could produce virulence just because waste products of the bacteria interact negatively with the host. It's just a coincidence, and tetanus isn't spread from individual to individual because, well, it's not doing that. It's not that's not the goal here. Then you have short-sighted evolution, and that is when the virulence is really due to a population of bacteria or viruses that are just living in a human, and just so happens that in order to have the highest fitness, they evolve in such a way as it actually impacts the host very negatively. So an example here is poliovirus. It infects the digestive tract and causes individuals to have diarrhea, which normally spreads the poliovirus quite well, but it can also infect nervous tissue, and when it does, it causes paralysis or death, which is deadly for the virus. But before it kills the virus in the human, it's actually advantageous to produce those new virions that can kind of grow at a higher rate than the older virions. HIV-X is an example earlier. It infects naive T cells and often leads to the death of the host. But what's good for part of a population of bacteria is, in the end, bad for the host. And for a little while, the bacteria grow faster, but then the host dies, and they do too. And last, virulence can be caused by a tradeoff. So in this case, what you have is a virus wants to spread itself as well as possible, but in order to do so, it's going to be killing the host because it takes so much energy away from the host. So you got like Vibrio cholerae. This is very deadly. It causes about 15% of the deaths per infection. And it's also very waterborne. So it's waterborne because it gets itself into the water. So once it gets into a human, it has to get itself into the water. And it does so by basically making the individual have a lot of feces, have a lot of watery vomit, and just get everything everywhere, and just hopes it gets into the water supply to be passed on again. But that's often deadly to the human via dehydration. Then you get things like non-typhoid salmonella, which is passed on by meat products, and yeah, it's not spread very fast, but it also doesn't go through the water supply, and it's not as deadly as something like cholera. So insect-borne pathogens like malaria should be interested in disabling the host. The host goes prone, the mosquitoes attack with advantage, and boom, free blood meal, including malaria, that they can pass on to someone else. Airborne pathogens as well should be mildly virulent. So they should keep the host walking around and spreading. And the best ones are going to be the ones that are relatively asymptomatic, in fact. And the host might not even know they have it while they spread it everywhere. Last up, cancer. So cancer is essentially a population of cells within the greater population of cells known as the human body. And these cancerous cells can also evolve. And what they evolve is, if they evolve a way to reproduce faster, then that is good for their own individual fitness. Their direct fitness is greater because the direct number of cells that they give rise to is, well, greater. So the indirect fitness of other cells staying alive goes down, but oh boy, direct fitness. And you get a population of cells that is essentially separate from the human body. It's living on its own, doing its own things. And if they can lose division control and divide faster, then they grow even faster. If they don't respond to apoptotic signals, they get a longer lifespan. So you see, even within that population of cancer cells, there's competition to be the fastest spreading cancer cell. And you have things like genome duplication, where it increases the number of potential mutations by increasing genome size. And of course, cells that avoid the cisplatin or other chemotherapies are going to grow even faster. So anything that increases the rate of growth of a cancer cell is going to be a positive for cancer. All right, so check. With a flu shot, you got in 2020, protected from viruses in 2022. I don't have the flu. Why or why not? One population is using antibiotics, the other uses vaccines. Which one's going to have is resistance? A new disease in resistance. I wrote this one back in 2019. It sounds a lot like COVID, but trust me. I wrote this in 2019. It would be much more lethal because the individual couldn't really respond to this very well, but the virus could actually become more pathogenic and spread faster, or less pathogenic and spread more, or it can spread faster either way, really. So 9 days post-infection, that would be absolutely tragic. Anyway, next we're going to discuss the human conditions and how they're affected by evolution.